Welcome back again to Cancer is a Mitochondrial Metabolic Disease Basics. We've already talked about glucose and we've already talked about glutamine, but does cancer only use glucose and glutamine as fuel? I think this begs the question, do other amino acids feed cancer too? This is where cancer metabolism gets a little more complicated and often misunderstood. Today, we're going to look at the evidence and separate real metabolic vulnerabilities from unnecessary dietary fears. So without further ado, let's get into it. So the question I want to answer in this video is, do other amino acids feed cancer? What the evidence actually shows. So I'm going to start out with this paper that was published in January of 2020, and it's titled Amino Acids in Cancer. And it says here that over 90 years ago, Otto Warburg's seminal discovery of aerobic glycolysis established metabolic reprogramming as one of the first distinguishing characteristics of cancer. The field of cancer metabolism subsequently revealed additional metabolic alterations in cancer by focusing on central carbon metabolism, including the citric acid cycle and the pentose phosphate pathway. Recent reports have, however, uncovered substantial non-carbon metabolism contributions to cancer cell viability and growth. Amino acids, nutrients vital to the survival of all cell types, experience reprogrammed metabolism in cancer. This review outlines the diverse roles of amino acids within the tumor and in tumor microenvironment. Beyond their role in biosynthesis, they serve as energy sources and help maintain redox balance. In addition, amino acid derivatives contribute to epigenetic regulation and immune responses linked to tumor genesis and metastases. Furthermore, in discussing the transporters and transaminases that mediate amino acid uptake and synthesis, we identify potential metabolic liabilities as targets for therapeutic intervention. And I cut out a couple of pieces of the paper here. The first one is I put cancer, how tumors hijack basic building blocks. And it says here, cancer changes how the body uses amino acids, the building blocks that all proteins are made of. A better understanding of these changes could lead to new cancer therapies. Cancer was already known to alter the body's sugar metabolism to feed extra energy to fast-growing tumors. Recent reports have revealed that cancer also rewires amino acid metabolism. Ji-Yeon Kim at the University of Illinois at Chicago, USA, and coworkers have revealed how cancer co-ops amino acids. They report that tumors use amino acids as an energy source and antioxidant precursor to balance their production of toxic reactive oxygen species. Amino acids are also instrumental in annotating the epigenetic code to enhance or suppress expression of tumor-related genes. The review illustrates a promising new approach to cancer therapeutics. And it says in the introduction, the nearly century of progress in cancer metabolism continues to impact our understanding of oncology. Each decade has yielded new discoveries and engendered additional avenues of research and approaches to therapy. Classically, cancer metabolism has focused on central carbon metabolism, including glycolysis and the TCA cycle. However, new studies have shed light on the important role of amino acids in cancer metabolism. Amino acids also serve the essential purposes of redox balance, energetic regulation, biosynthetic support, and homeostatic maintenance. The wide breadth of metabolic activities has made amino acid metabolism increasingly popular in cancer research. And it gives a couple of graphics in this paper and some associated explanations next to the graphics. So here in figure A, top left-hand corner, cells can either import amino acids from the extracellular environment or synthesize them inside the cell. Amino acid transporters are required to transfer them across the plasma membrane, shown in A, and transaminases are required to generate amino acids from sugar precursors and nitrogen from other amino acids, shown in B, bottom right. Amino acid transporters and transaminases contribute to metabolic reprogramming in cancer through manipulation of the amino acid pool inside and outside cancer cells. Their impact on tumor microenvironment is associated with tumor aggressiveness. Some induce tumor cell growth and expansion through the activation of signaling pathways, mTOR activation, or inactivation of tumor suppressors, while others result in apoptotic cell death. So what I've been getting at to this point is that amino acids are needed for all cells, for raw materials and building blocks. But the question that I was going to ask in this video, the central question is, do these amino acids actually feed cancer from a bioenergetic perspective? That is the question. So you may or may not be aware of is there are amino acids that are considered glucogenic, and there are amino acids that are considered ketogenic. Basically, either amino acids that break down into glucose and they'll be metabolized through the glucose pathways, or they will be broken down into ketones and they'll be broken down to ketogenic pathways or to TCA intermediates as shown here. And I'm going to use a couple of Seyfried's papers to help answer these important questions about amino acids. The first one was published in January of 2018, and it's titled Mitochondrial Substrate Level Phosphorylation as Energy Source for Glioblastoma, Review and Hypothesis. And it says here, literature is reviewed suggesting that the succinyl-CoA ligase reaction in the TCA cycle, or the tricarboxylic acid cycle, can substantiate sufficient ATP through mitochondrial substrate level phosphorylation, MSLP, to maintain GBM, or glioblastoma multiforme, growth when OXPHOS is impaired. Production of high energy phosphates would be supported by glutaminolysis, or the breakdown of glutamine, or the metabolism of glutamine, a hallmark of GBM metabolism, through the sequential conversion of glutamine to glutamate, glutamate to alpha ketoglutarate, and to succinyl-CoA, and lastly, succinate. It later says in the paper that other potential metabolic fuels in the tumor microenvironment, for example, acetate and branched chain amino acids, are either not present in sufficient quantities to drive growth through fermentation or exert metabolic effects that are yet to be understood. While amino acids other than glutamine could be fuels, they require energy for preparation prior to their catabolism or their breakdown. I'm going to really hit that in a second with pictures so it makes sense. So it says in this paper later on that there are a number of metabolites that catabolize or break down towards the citric acid cycle through succinyl-CoA, thus leading to ATP or GTP formation through succinyl-CoA ligase. Our review of the relevant biochemistry, however, shows that only glutamine catabolism or breakdown can yield a net positive ATP production through this mitochondrial substrate level phosphorylation. And it says later, except for glutamate and glutamine, however, the catabolism of all other metabolites expend one or more high energy phosphates during metabolic interconversions before becoming succinyl-CoA. Support for mitochondrial substrate level phosphorylation could also come from the conversion of glutamate, such as proline and arginine, other amino acids, or from metabolites that catabolize towards alpha ketoglutarate, a key intermediate in the TCA cycle, such as glycine, serine, sarcosine, dimethylglycine through pyruvate, and fatty acids or leucine through acetyl-CoA. However, the concentrations of these metabolites in the plasma or in the microenvironment are far lower than that of glutamine, and therefore unlikely to make major contributions to this mitochondrial substrate level phosphorylation, or the making of energy through broken mitochondria through this fermentation pathway and using mitochondria. So what we have in this picture effectively is we have basically all of these amino acids, whether it be serine, glycine, sarcosine, glutamine, histidine, proline, arginine, et cetera, valine, isoleucine, et cetera. All of these amino acids are broken down in various ways to cycle into the TCA cycle or the Krebs cycle or the citric acid cycle. There's multiple names for the same pathways. The key finding is twofold. Number one, only glutamine metabolism, or the breakdown of glutamine, can yield a net ATP gain, which means that all these other amino acids can break down to make energy. It's just that it costs energy to make energy in a way that is either net zero or net negative. So therefore, it would not prolong the survival or the thriving of these cancer cells. In addition to that, if that wasn't enough, unfortunately, some of these other metabolites that potentially could break down to make energy, not only do they cost energy, but they're not found in sufficient quantities in the tumor or the tumor microenvironment in order to support the growth, unlike glutamine, which has a much higher concentration. This paper was published in 2024, and we've seen this recently. And it says the results showed that glutamine was a source of ATP production or content irrespective of oxygen, No other amino acid could replace glutamine in sustaining ATP content and viability. So what they did is they took these cancer cells and they added one at a time different amino acids, and they looked at 24 hours and at 48 hours what was keeping these cancer cells alive when glucose was present or when glucose was absent. And so we see here that this is the glucose negative or the glucose absent medium. And you see here at 24 hours that only glutamine and glutamate are keeping them above the normal amounts. And then at 48 hours, really only glutamine is able to make up for the lack of glucose in these cells. All other amino acids have diminishing returns and will ultimately lead to energetic cell death and failure. And then they have here basal glucose that's there, right? They actually add glucose back in. And you see here that again, glutamine is what is keeping these cancer cells not only alive, but thriving. It is both glucose and glutamine that are the main fuels for cancer. Does that mean that cancer is not utilizing these amino acids in other ways as raw materials for growth? No, that is true. But in terms of keeping the cell alive, glucose and glutamine are the two and major and only fuel sources for cancer. And I pulled this paper that was published in February 2023, and I didn't show the abstract because I don't think it's important, but it's called The Concentrations of Plasma Amino Acids and Neurotransmitters in Participants with Functional Gut Disorders and Healthy Controls. And I'm mostly concerned about the healthy controls. And I just wanted to show kind of the relative differences in the concentration of different amino acids in the bloodstream. So we see things like, I don't know, citrulline, which is 35, or histidine, which is 59, or asparagine, which is 56, or aspartic acid, which is 6, whereas we've seen glutamic acid at 54, serine at 113, glycine at 249. But again, glutamine is the highest amount of the amino acids in the bloodstream at 585.5. The next highest was like alanine at 402. There's a couple in the 200s. Here's leucine at 144. But glutamine is at least 2, if not like 10-fold higher than every other amino acid. And then, as we saw earlier in the video prior to this, whether or not glutamine is used to feed cancer, cancer will utilize glutamine preferentially 10 to 100-fold more than any other amino acid. And it shows because it's the only amino acid that's been shown when glucose is present or absent to keep these cancers alive. It's the only alternative fuel source that will actually yield a net positive energy result. So glucose and glutamine are the primary fuel sources for cancer, but other amino acids can be utilized as raw materials, just like fatty acids can and ketones and other things. But in terms of energetics, bioenergetics, glucose and glutamine are the only two. If you like videos like this, please like, share, and subscribe. And until next time.